o'clock. Okay, today in the second session of the embryology class, we are going to talk about the formation of the cardiac septa. Formation of the cardiac septa. The major septa of the heart are forming between the 27th and 37th days of the embryonic period. Okay, 27th to 37th days of the embryonic period. And there are some ways of forming the septums. We will talk about the general forms of septum formation, and then we will talk about each septum one by one in detail. Okay? One of the ways of formation of the septums, or one method, is involving two activity growing masses of tissue. These two tissues, look at here. For example, in a lumen, okay, we have two growing masses of tissue which are growing to each other's side, and finally they are fusing to each other. They are approaching to each other and fusing to each other. So the lumen of the tube will divide into two parts, 1 and 2. Okay? Some masses are growing in the tissue and connecting to each other, so the lumen will be divided into two parts. The next way of forming the septums of the heart is that there is just one mass in one side of the lumen. This mass is growing to the other side. Okay? So, there is just one mass which is growing to the other side. This is the other way. We call these growing masses, yes, please ask your question.
What about it's either by the part B or either by part C?
What is the question?
The growing of the septa is either by the part B, the picture B, or by C?
In some parts we have the type B formation, and in some other parts we have type C. We will talk about each septum in detail, one by one. Now I'm just talking about the general method. Okay? You're welcome. So, we call these growing masses the endocardial cushions. So, these growing masses are called the endocardial cushions. These kind of endocardial cushions are forming in the matrix of the cell. Okay? Formation of these kind of tissue masses, which are called the endocardial cushions, depends on the synthesis and deposition of the extracellular matrix. Look at here. We can see that in each lumen we have one layer of endocardium. After that we have the extracellular matrix, or ECM, extracellular matrix, or ECM. And then we have the myocardium. Okay? We have the myocardium. So, by the synthesis and deposition of the extracellular matrix, we can see that these endocardial cushions are forming, like this part. These endocardial cushions are forming, and they are proliferating and migrating to the side of each other or to the other side, and then they are dividing the lumen into two parts. These endocardial cushions are developed in atrioventricular and conotruncal regions. The septum means a valve in a lumen. A valve in a lumen which divide the lumen into two parts. Yes. Okay? So, these endocardial cushions are forming the septums, the atrioventricular and conotruncal region. So, they are forming the atrioventricular and conotruncal. The pointer isn't moving. Okay. Okay, the conotruncal region. And also, in these locations, they assist in the formation of atrial and ventricular septa. Listen, please. In the atrioventricular and conotruncal regions, the endocardial cushions are forming atrial and ventricular septa, or the atrioventricular canals and valves, and also aortic and pulmonary canals. So these are forming these parts. The other form of the formation of the septums is that the two parts besides the septum are growing faster. Okay? Instead of the endocardial cushion growing, instead of the growing in this ridge, the two parts of the lumen which are located beside the ridge are growing. But in the region, in the area of this ridge, there is no growing. In the area of this ridge, there is no growing. So, this part will form a septum. So, there are two forms. In one form, we have the endocardial cushions in the ECM or extracellular matrix, which are growing to the side of each other. Okay, or we have just one cushion. In the other form, there is an area in the lumen which do not grow, but the lumen which are located beside this area are growing. So, this part is forming a septum in the lumen. Okay, these parts are forming a septum in the lumen. Here, as we have seen, we can see the proliferation and migration of the cell in the ECM or extracellular matrix, which are forming the endocardial cushions, and they are dividing the lumen into two parts. In the second form, in this part, in this form of septum formation, there would be just a foramen like this in the lumen, and the lumen would not divide into two parts completely. Listen, the septation and the division of the lumen into two parts will not occur completely. We will just see a foramen in the lumen. Okay, a duct, we can say, is remain in the lumen. So this is the general form of the septum formation. The first septum, I'm talking about the third slide. Third slide. Yes or no? Yes, I think the third one. Yes. If you can't see the slides, I will explain which slide I'm talking about. Okay. At first, we are going to talk about the septum formation in common atrium. You remember that in the cardiac tube, we had just one part. We call that the common atrium. Look at here like this. Near the sinus venosus, we had the atrial part of the cardiac tube, and it was just one common cavity which was called the common atrium. At the end of the fourth week, just a sickle shape, I will show you that, okay? A sickle-shaped crest grows from the roof of the common atrium. Look at here, in this picture, we can see the common atrium, and this is the roof of the common atrium. No problem. Okay, from the roof of the common atrium, a sickle-shaped crest will grow. We call this crest the septum primum. This crest is called septum primum. The two limbs of this septum are growing toward the inferior side. The two limbs of this septum are growing into the inferior side, and they are growing toward the atrioventricular canal. Look at here, we have the atrioventricular canal. Can you remember that we had a common atrium and then we had the ventricular part, the left ventricle and then the bulbus cordis. Okay? Here also we can see the ventricular part, and this is the common atrium. So this septum primum is growing toward the atrioventricular canal. The opening between, listen please, the opening between the lower part of this septum, the lower part of the septum, and the endocardial cushions in the atrioventricular canal is called the ostium primum. So, at the time of septum formation in the common atrium, we can also see some endocardial cushions in the atrioventricular canal at the same time, near the fourth week. Okay? At the time of the fourth week, we can see the septum primum in the common atrium cavity and also some endocardial cushions in the atrioventricular canal. The septum is growing toward the endocardial cushions, and the foramen which is formed between the lower limb of the septum primum and the endocardial cushions is called ostium primum. Look at here, we can see in the sagittal plan. We can see the septum primum, and here also we have the ostium primum. These are also the endocardial cushions in the atrioventricular canal. Okay? The endocardial cushion and also the septum primum are growing toward each other, and they want to, what are you talking about? They are growing toward each other and they want to close the ostium primum. They are growing to the side of each other and they want to close the ostium primum. But before the closure of the ostium primum, we can see some cell death in the superior part of the ostium primum here. At the time of the closing of the septum primum, the ostium primum, and before its closure, some cell death procedures are happening in the upper portion of the septum primum in this part. These perforations are connecting because they want to form another ostium. Okay? Because they want to form another ostium. No, where are the coronary arteries? We are talking about the atrium. Okay, don't be confused. We are talking about the septum formation in the atrium. Interatricular septum. These perforations are connecting to each other at this time and forming ostium secundum. Look at here. The ostium primum is closed because the septum primum is growing downward and also because the endocardial cushions are growing toward each other at this part. But before its closure, there would be another ostium in the septum primum which is called ostium secundum. Okay, clear? Ostium secundum. The blood can pass through these ostium and enter to the left atrium from the right atrium. Now, listen please. The septum primum is forming to separate the right and left atrium. The septum primum is forming to separate the left and right atriums. There is a hole in the septum primum which is called ostium primum. The ostium primum is closing by the growing of the septum primum and these endocardial cushions. But at the superior portion of the septum primum, there is another hole which is called ostium secundum. Okay? The blood will enter to the left atrium from the right atrium through ostium secundum. When the lumen of the right atrium is expanding, look at here, also we can see the ostium secundum here. And these are the endocardial cushions. These are the endocardial cushions, okay, growing toward each other. And here also we can see the ostium secundum. But at the time of the right atrium expansion, listen please, this part is so important and so hard. You have to understand. At the time of expansion of the right atrium, another septum is forming from the roof of the right atrium. Okay? Another septum, another crescent shape septum or sickle shape septum is forming, and at the right side of the septum primum, another septum is forming in the right side of the septum primum. This septum is called septum secundum. This part is called septum secundum. The septum secundum never forms a complete partition in the atrial cavity. The septum secundum never forms a complete partition in the atrial cavity. Its limb is growing and extending downward to the septum in the atrioventricular canal. Like septum primum, the septum secundum is also growing to form to the side or toward the endocardial cushions in the atrioventricular canal. At the time when the sinus venosus is opening into the right atrium, listen please, at the time when the sinus venosus is opening into the right atrium, it has two valves, right venous valve and left venous valve. Do you remember these parts? Right and left venous valves. There is a septum on the superior portion of these valves which is called septum sporium. Here we can see septum sporium. Okay? When the sinus venerum is expanding in the right atrium cavity, the left venous valve, I mean this valve, the left venous valve and septum sporium are connecting into the septum secundum. Look at here, they are growing into the left side of the atrium and attaching into the septum secundum. Here, they are attaching into the septum secundum. The opening left by the septum secundum is called foramen ovale. Foramen ovale. Look at here. This is the septum secundum, which is going downward and receiving septum sporium and left venous valve. The opening which is remain in this part is called foramen ovale. Here also we can see the foramen ovale, an oval shaped foramen or opening. Okay, clear? And when the upper part, why don't you understand? Look at here, you understood septum secundum, okay? This is the septum secundum. This valve, I mean left venous valve and septum sporium are also growing and moving toward the septum secundum and attaching to its parts. I said that the septum secundum cannot never completely close the atrial cavity or separate the atrial cavity. There would remain a foramen in its downward portion. Okay, this foramen is called foramen ovale. This opening is called foramen ovale. And when The upper portion, listen, when the upper portion of the septum primum disappears, this part is disappearing because of cell death and forming ostium primum. When this part is disappearing, the remaining part of septum primum, I mean this part, the remaining part of septum primum is called valve of the oval foramen. The remaining part of septum primum is called valve of the foramen oval. Okay? Slide number, I think. Yes, 5. Okay? So, in the embryonic development period, the blood is passing through foramen oval and this foramen and entering to the left atrium from the right atrium. After birth, when the lung circulation is start, listen please, when the circulation of the lungs start or begins, the pressure in the left atrium will increase. So, the valve of foramen oval will close this foramen. Okay? Yes, when the pulmonary circulation begins. No, I said that it is never closed. I mean, it is never growing downward to reach to this part. Okay? But on the other side, we have septum primum. It works as a valve. Yes. But after birth, when the pulmonary circulation and the function of the lungs begin, the pressure in the left atrium will increase, so this valve will close the foramen oval. This valve will close the foramen oval. If this foramen oval remain open, or for example just a small foramen remain open in this part, we call that prob patency. We call that prob patency. In 20% of cases, the fusion of the septum primum and septum secundum is incomplete after birth. In 20% of cases, the fusion of the valve of foramen oval, I mean septum primum, and septum secundum will be incomplete, so there will remain just a cleft between these two parts. We call that prob patency. But it is not a problem, a big problem. Okay? Any question about this part? Look at the, I said, sickle-shaped septum. Look at here, this is septum primum. If you can't hear my voice and also you can't see the slides, please exit the class and enter again. I can't do anything. Yes. Yes, it's propatency. This is not my problem from my side, okay? If you can see the slide and also you can hear my voice, this is the problem of your network. Exit the class and enter again. I will refresh the slides to see if you can see the slides or not, okay? Let me. I will uploading my file one more time. Now we are going to talk about the formation of the left atrium and pulmonary veins. Formation of the left atrium. So we talked about the septum formation in the common atrium. Now we have two atriums, right and left. Okay? Now what about the formation of the left atrium and the pulmonary veins? I will wait. The file is uploading. Wait, please. Okay, my file is uploading. Can you see the slides?
Khanam, why don't you use the Skyrim instead of Adobe Connect?
We
It's
can't
very better.
use Skyrim because we don't have any link now. Okay,
Can you ask the university to have it tomorrow?
for the Skyrim, is it better? Because I have many network problem with the Skyrim application.
It needs less internet than Adobe Connect, or less issues.
I don't know if you ask, please, no problem.
We can't contact the university. You are our way to contact it.
Okay, 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 okay. Now listen, please. I'm talking about slide number 6. In this slide, you can see the sickle cell septum primum. Here. Today now. Yes, please. I beg you, please. In this part, we can see the septum primum, and here we can see the sinus venosus. Here also we can see the septum primum and the cellette on the upper portion, which are forming the septum, the ostium primum. This is also the septum secundum, and these are the endocardial cushions. These are the endocardial cushions in the atrioventricular canal. And in this session, slide number 8, you can see the complete formation of septum secundum and also septum primum, the vulva foramen oval, and this is the foramen oval itself. Now about the formation of the left atrium and the pulmonary vein. At the time when the sinus venosus is connecting to the right atrium and expanding, in the left atrium also we can see some expansions. The mesenchyme at the caudal side of, or we can see at the roof of the common atrium, okay, this is the mesenchyme at the caudal side of the dorsal mesocardium. Do you remember the dorsal mesocardium? The mesocardium which suspends the cardiac tube in the precardial cavity. The mesenchyme at the caudal end of this mesocardium is growing and proliferating in the precardial cavity. It begins to proliferate, okay? At the time which septum primum is growing into the side of endocardial cushions, in the side of endocardial cushions, we can see that this proliferating mesenchyme is forming dorsal mesenchymal protortion. Dorsal mesenchymal protortion. Dorsal mesenchymal protortion, or DMP. This dorsal mesenchymal protortion is growing with septum primum toward the atrioventricular canal. Look at here, we can see it here. It is growing at the side of atrioventricular canal. Inside this mesenchymal protortion, we can see the pulmonary vein, the opening of pulmonary vein. So, inside the DMP, we can see the opening of the pulmonary vein. This developing pulmonary vein is positioned in the left atrium by the movement of DMP. At first it is in dorsal mesocardium, then it is growing downward to the side of atrioventricular canal, and we can see the opening of the pulmonary vein inside these parts, inside this mesenchyme. Eventually, the remaining portion of DMP at the tip of septum primum, I mean this part at the tip of septum primum, contributes to the endocardial cushion formation in the atrioventricular canal. It is coming downward and contributes in the formation of the endocardial cushions in the atrioventricular canal. The stem of the pulmonary vein remains in the dorsal side of the left atrium and forming and dividing into 4 parts, 2 pulmonary veins in the right side and 2 in the left side. They are expanding into the left atrium and forming the 4 pulmonary veins into the atrial chamber. Okay, so generally we have dorsal mesocardium. At the caudal side of dorsal mesocardium, we have a part with the name of dorsal mesenchymal protortion. This protortion is proliferating and growing toward the atrioventricular canal with the septum primum. The remaining portion of this mesenchyme is contributing in the formation of the endocardial cushions at the side of atrioventricular canals, and also the pulmonary vein is expanding in this mesenchyme. At this time it is dividing into 4 parts and forming the pulmonary veins. Forming the pulmonary veins. Okay, understood? Okay. Look at here. In this slide, slide number 10, we can see right venous valve and left venous valves, which are expanding into the right atrium and forming the smooth valve part of the right atrium. And also here we can see the pulmonary veins, the DMP in the left atrium, which are forming the smooth valve part of the left atrium. So the smooth valve part of both atriums are forming by sinus venosus and pulmonary veins. The row valve part of each atrium is called the auricle and are the remain parts of the common atrium. Slide number 11. Septum formation in the atrioventricular canal. Septum formation in the atrioventricular canal, slide number 11. At the end, slide number 10 is not showing DMP, but it's here in this part. Near the pulmonary vein opening. It hasn't been shown in this picture, in this slide. Slide number 10, I mean. Now, slide number 11. Okay, we want to see how the septum is forming in the atrioventricular canal. At the end of the 4th week, four atrioventricular endocardial cushions are appearing. As we said at the time when the septum formation is happening in the common atrium, also at the side of atrioventricular canal, 4 endocardial cushions are forming, okay, at each side. Look at here, these endocardial cushions, superior, inferior, left and right and left endocardial cushions. These endocardial cushions are growing to each side, toward each other. The growing in the superior and inferior endocardial cushions is more than the other cushions, so they are growing toward each other and dividing the lumen into 2 parts. Look at here. This is this foramen in this part, in this picture. We can see the superior and inferior endocardial cushions. They are growing toward each other and dividing the atrioventricular canal into 2 parts, right atrioventricular and left atrioventricular canals. At the first, the atrioventricular canal gives access only to the primitive left ventricle. If you remember, we could see that the common atrium was connected to just the left ventricle. Do you remember? But we said with the formation of the atrium, right and left atrium, and their expanding, the bulbus cordis is moving into the middle side of the cardiac tube. They are shifting this part into the left side. So, at that time, the blood is entering, instead of entering just to the left ventricle, the blood is also entering to the right ventricle, to the proximal part of the bulbus cordis, the right ventricle. So, near the end of the 5th week, we can see that the atrioventricular canal is separating the 2 atriums from 2 ventricles, right and left to right and left ventricles. After the connection of these endocardial cushions, we can see the right and left endocardial canals. So, the blood is passing from these atrioventricular canals and entering to each ventricle from the left atrium to the left ventricle and from the right atrium to the right ventricle. As you pay attention, we have also some lateral endocardial cushions. We have also some lateral endocardial cushions which are appearing in right and left borders of the canal, but their proliferation is much less than the superior and inferior ones. They are contributing in the formation of the tricuspid and mitral valves. They are contributing to form the tricuspid and mitral valves. I will explain later. Any question in this part? Now, we can, in this slide number 12, we can see the contribution of the cells to endocardial cushions. Look at here, this is a beautiful picture of these happenings. We can see the cardiac tube. This is the aortic sac. Listen please. We can see the dorsal mesocardium and also the primitive cavity. This is the transverse sinus. Do you remember? This is the transverse sinus. The endocardial cushions here. This is the aortic sinus. In this part, we can see the outflow tract of bulbus cordis, and this is the ventricle, the left ventricle, common left ventricle. Here is the right ventricle. And also we can see the endocardial cushions in the atrioventricular canal. This is the common atrium. Here we can see DMP or dorsal mesenchymal protortion, which I said it's in the caudal side of the dorsal mesocardium for the formation of the pulmonary veins. And also here we can see the sinus venosus. And also in this picture we can see the superior and inferior endocardial cushions. These are the lateral endocardial cushions. The superior and inferior endocardial cushions are also called dorsal and ventral cushions. Superior and inferior are also called dorsal and ventral cushions. Slide number 13. Formation of the atrioventricular canals and chorda tendina. After the formation of the right and left atrioventricular canals and the endocardial cushions, each atrioventricular orifice is surrounded by the proliferation of mesenchymal tissue. Look at here. In slide number 11, we can see the endocardial tissue on each side of the orifice. These are proliferating, okay? After that, after this proliferation, when the blood stream is flowing inside each ventricle, when the blood is flowing inside each ventricle and entering to the ventricles from atriums, the epithelial tissue on the ventricular surface of this proliferation, the mesenchymal tissue the mesenchymal tissue becomes fibrous. This is a ventricle. This is the ventricle, okay? After the blood flow into each ventricle, we can see that the myocardium, the tissue of the myocardium is becoming fibrous in some parts and forms the atrioventricular valves. Look at here. This is fibrous part, and we can see it forms atrioventricular valves. These valves are remain attached to each ventricular valve. These valves are attached to the ventricular valves by these tendina, which are called chorda tendina. The other parts of the muscle in the ventricle are degenerating because of the blood flow. Because of the blood flow, we can see that the muscle is degenerating. Yes, and the chorda tendina are connected to the muscles of the ventricular valves via the papillary muscles. Okay? So the blood flow is forming the last formation of the atrioventricular valves. The muscle is degenerating, and also a remaining part is its tissue is a fibrous form, which are connected to the papillary muscles via chorda tendina. Yes, the blood flowing is degenerating the surface, the muscles, because it has high pressure. It has high pressure. Think that, for example, you have a loose tissue in some part and you are flowing water or everything to that part. So degeneration is happening. In ventricles also we have this. This part is forming the atrioventricular valves, the tricuspid valve in the right side and bicuspid or mitral valve in the left side or left atrioventricular canal. Bicuspid valve and tricuspid valves in the right and left sides of the heart. Right and left sides of the heart. So in this way, the atrioventricular canals and valves are so forming. Now, before a rest, let's talk about some of the defects which may happen in the heart because of the septums. Okay. Some of the happenings which may affect the heart formation and the septum formation in the embryonic period can be, for example, some viruses like rubella, and also some drugs, some like thalidomide. These may have some effects in the cardiac formation and forming some heart defects which are common. Heart defects which are common. Okay? They are targeting, and also, for example, some drugs, some like Accutane or ARA, Accutane. The alcohol also may affect. the alcohol, and also some diseases in the, or it could be better, some matter now. Diseases like diabetes, high blood pressure, and these will make some problems for the baby's heart. And also there are some genetic points which may be affected, and also after them we have some heart defects. Some of the important genes in the formation of the heart are, for example, NKX 2.5. NKX 2.5. Because maybe rubella virus will pass through the blood and affect the embryo. And a book for clinical, Longman is explaining the clinical correlates. Longman is explaining all these clinical points. Okay. I said that one of the important genes in this process is NKX 2.5 and also TBX5. These genes are connected to each other and also present in the heart formation and also in the limb formation, upper limb formation. Because of that, the defects that may be related or correlated to these kind of genes are called heart-hand defects. Because there is a gene in the formation of the cardiac septum and the upper limb, and if there is a defect in the function of this gene, the problems which may happen are called heart-hand syndromes. For example, one of these syndromes is Holt-Oram syndrome. Holt-Oram syndrome. It has some upper limb abnormalities and also some heart defects, for example, ASD or atrial septal defects. They have some defects like ASD and also some abnormalities in the upper limb. And also we call them heart-hand syndromes because of these genes. Another defect in the heart is hypertrophic cardiomyopathy, which will happen because of some defects in the myocardium. The myocardium will be hypertrophic. These may happen in sudden death in the athletes or the general population, okay? Because the myocardium of the heart is hypertrophic and it needs much blood in the athletes, they will occur sudden death, or also in some general population. It's because of some myocardial disarray, and the cells are proliferating, the myocardial cells are proliferating very much, and this condition will happen. We have also ventricular inversion. It's a name of a gene. TBX5, it's a name of a gene which is contributing in upper limb formation and also the heart. It's abbreviation if you mean, I don't know exactly. We have some ventricular inversion. Ventricular inversion is a defect in which, look at here, in part A we can see hypoplastic right heart syndrome, but now we are going to talk about ventricular inversion. Okay, ventricular inversion is a defect in which the morphology of left ventricle is on the right and connects to the right atrium. Okay, so the left ventricle is connected to the right atrium through the mitral valve. Yes, the upper limb and heart, septums of the heart. So, the left ventricle is connected to right atrium through the mitral valve, and the right ventricle is connected to the left atrium through tricuspid valve. It is called ventricular inversion. This defect is also called L transposition of the target arteries. The ventricular inversion is also called L transposition of the great arteries, because the pulmonary artery exits the left ventricle and the aorta exits the right ventricle, the morphological right ventricle. They have these problems. The arteries are in their normal position. The arteries are placed or located in their normal position, but they are existing from reversed ventricles. The ventricles are reversed. And this will happen through the laterality process when the laterality pathway is forming. Yes, transposition means replacement. No. Transposition does not mean replacement, because the arteries are located in their right side, but they are existing from the reverse ventricles. They are exiting from the reverse ventricles. The other form of defect is hypoplastic right heart syndrome and hypoplastic left heart syndrome. In this kind of defects, they are rare defects. They cause underdevelopment of the right or the left side of the heart. Hypoplastic right and hypoplastic left side of the heart. We have undevelopment in these parts of the heart. On the right side, look at here, we can see that the ventricle, the right ventricle, is very small. In this part, it is very small. The right ventricle is very small. The pulmonary artery also is affected and may be atretic or stenosed. The pulmonary artery is atretic. The atrium is also maybe small. But the left ventricle and left atrium are very dilated. On the other part, the hypoplastic left heart syndrome, we can see that the left ventricle and left atrium are small and hypoplastic, and also the aorta. But the other side of the heart, right atrium and right ventricle, are very dilated. In both kind of these defects, the ductus arteriosus will remain open. No different. They both have problems. They have different problems, okay? But the ductus arteriosus will be open, will remain open, and the oxygenated and deoxygenated blood are mixing. These are mixing. The other defect, no, there is no other thing. As I know, there is no Q. The other defect in the septum formation is ASD, atrial septal defect. ASD is an incidence of 6.4 to 10,000 births. I will explain. When I'm explaining the septum formation in bulbus cordis, you will understand how will it happen, okay? I will explain, of course. Yes. And this is one of the defects in the atrial septum. Then, look at here. For example, here we can see ASD. It may happen by many ways. One of the most significant defects is the ostium secundum defect. In ASD, one of the most significant defects is the ostium secundum defect. Look at here, I mean the blue one. I'm slide number 15. If it has some defects, this will happen. So there would be a large opening between the left and the right atria. It may happen because of the cell death. It may happen because of the cell death. And also here we can see that, look at here, for example, in this part, the septum secundum has some problems, but in other form, it's because of expanded cell death, and we can see a big ostium primum. A big ostium primum in this side. The blood will enter to the left atrium from the right atrium, and also here we have a shunt. And the oxygenated and deoxygenated blood are mixing. Here we can see the septum secundum, absence of septum secundum. And this is the atrial septal defect. Look at here, we don't have septum secundum. The other, the most serious abnormality in this group is complete absence of the atrial septum. The most serious problem in this part is completely the absence of the atrial septum. We call that core tricular biventricular. I mean, the heart just have 3 cores. 1 common atrium and 2 ventricles. It has serious defect in the heart. So the most serious problem is the absence of the atrial septum. So we have the core tricular biventricular. It means the heart have just 3 chambers, 1 common atrium and 2 ventricles. In slide number 16, the other problem is the persistent atrioventricular canal. Persistent atrioventricular canal. Also, we have some other problems, for example, premature closure of the oval foramen. It means the oval foramen is closed sooner than the right time. Okay? And if the problem is happen in the endocardial cushions, there would be some defects. For example, one of them is persistent atrioventricular canal. It means the atrioventricular canal won't separate and divide two canals, and just we have one. Look at here. Here we have the endocardial cushions. In the persistent atrioventricular canal, these cushions have some abnormality and defects. And they are combined with a defect in cardiac septum. In this condition, we have also some cardiac septum. This septal defect has an atrial and ventricular component, separated by abnormal valve leaflets in single atrioventricular orifice. Instead of right and left, I mean tricuspid and mitral valves, we have this problem, okay? We have just one canal, atrioventricular canal. And also the endocardial cushions in the atrioventricular canal partially fuse. In some occasions, they are partially fused. So the defect is happening in the atrial septum, but the interventricular septum is closed. This defect is called the ostium primum defect. Ostium primum defect. So most of the time, this kind of defect has a problem in tricuspid valve. They have problem in repeat what? Repeat the persistent atrioventricular canal, you mean? Okay. If the endocardial cushions have some abnormality, these kind of defects will happen. For example, in persistent atrioventricular canal, you can see that these cushions won't grow toward each other and attach to each other. So the atrioventricular canal would not divide into two parts, and we have just one atrioventricular canal, not two. Instead of tricuspid and mitral valves, we have just one. Okay? And in the other defect, ostium primum defect, in this form of the defect, we have combined with a cleft in the anterior leaflet of the tricuspid valve. They have defect in the atrial septum. Look at here. We have two other kinds, tricuspid atresia here. This is the normal heart, and this is tricuspid atresia, slide number 17. In the tricuspid atresia, the orifice of the right atrioventricular canal is closed. So the right ventricle will be hypotrophic. The right ventricle will be hypotrophic. And we don't have the tricuspid valve. The ductus arteriosus will remain open. And also the foramen oval will remain open. Yes, the fusion will occur in the tricuspid valves and also the endocardial cushions. Also, this will happen in the mitral. Look at here, so we have hypotrophic right ventricle, open foramen oval, open ductus arteriosus, and mixing of the blood. We have also another anomaly with the name of Epstein anomaly in this part. The tricuspid valve is displaced toward the apex of the right ventricle. It is displaced of Epstein of the tricuspid valve. So we can see that the valves of tricuspid valve are attached to the apex of the right ventricle. As a result, there is expanded right atrium, a big right atrium, and a small right ventricle. The blood is entering to the pulmonary artery from this side, atrioventricular canal. I think there is no recovery for these kind of problems because they have many problems with each other. They are syndromes, so there is no recovery. Take a break for some minutes and then I will continue my lesson. Just for 5 minutes because we don't have time, okay? And in these 5 minutes, I will give you the link for the attendance. I'm explaining slide number 18, I think. 18. Yes. Septum formation in the truncus arteriosus and conus cordis. Here, after explaining this part, you will understand the transposition of the big vessels, okay? Look at here. During the 5th week, some pairs of opposing ridges appear in the truncus. Some pairs of opposing ridges appear in the truncus. These ridges are called truncus swellings or truncus cushions in the truncus arteriosus. These are placed or located on the right superior, look at here, right superior wall and left inferior, right superior truncus swelling and left inferior wall. Because of that, we call them opposing swellings, right superior and left inferior. Okay, we have these two swellings. They are growing distally and to the opposite side. I mean, the right superior truncus, this one, truncus swelling, the right superior truncus swelling, growing distally and to the left side. The left inferior truncus also growing distally and to the right side, in the opposite direction, distally and to the right side. At the time of growing these parts, they are growing toward the aortic sac. Okay, they are growing toward the aortic sac. Here we can see aortic side. Number 18, slide number 18. And also, these two swellings are twisting. Look at these pictures, A, B, and C. They are twisting also around each other and forming a spiral septum. They are twisting around each other and forming a spiral septum. And they are fusing to each other, like part B. The purple and the yellow colors are twisting around each other and fusing. They twist because they want to connect the aorta to the left ventricle and the pulmonary trunk to the right ventricle. If they do not twist, this won't happen. So we have the transposition of the big vessels, big arteries. So they are twisting around each other and forming a spiral septum. This ridge, after the completion of the fusion, this ridge and this septum is forming aorticopulmonary septum. Aorticopulmonary septum. This septum grow toward aorta. That's why, am I right or not? Yes, they are growing towards the aortic sac. So this aorticopulmonary septum is dividing the truncus arteriosus into the aortic and pulmonary canal. Into the aortic, look at here, the red arrow, and the blue arrow, the pulmonary canal. These cushions are also growing into the atrioventricular canals and septum. They are also growing at the other side, I mean toward the endocardial cushions in the atrioventricular canal, in both sides. And connecting to this, look at here, proliferation of the inferior atrioventricular cushion. And at the end part, they are connected to the interventricular septum. Okay? At the time, when the truncus arteriosus swellings appear, also some similar cushions and swellings develop along the right dorsal and left ventral wall of the conus cordis. Right dorsal and left ventral of the conus cordis. So we have these cushions in the truncus arteriosus and conus cordis, two parts of the bulbus cordis, two distal parts, middle and distal part. So in the conus cordis also we have dorsal and right dorsal, I mean this one, and left ventral, these two, left ventral walls of the conus cordis. These conus swellings also grow toward each other and distally to the truncus septum. These are also growing toward each other and growing toward the swellings of the truncus arteriosus to complete the septum. So we have two parts of the outflow, one for the aorta and one for the pulmonary trunk. Was it clear? We have endocardial cushions in the truncus arteriosus and conus cordis, I mean the middle and the distal part of the bulbus cordis. We have to finish, so don't be tired. These cushions are twisting, growing toward each other and twisting and forming a spiral septum in this part, and also growing to the side of atrioventricular canal and forming the canals for aorta and pulmonary arteries. If this twist won't happen, there would be some problems, like, for example, as I said, the transposition of the big vessels. We have some cardiac neural crest cells. These neural crest cells are, or as you remember, are originating in the edge of the neural folds of the neural tube. Then they are migrating from this side and through the neck, okay, and through this region, or pharyngeal arcs, number 3, 4, and 6, and come into the heart region or cardiac region. They are contributing in the formation of the cardiac septums. So the neural crest cells are contributing in the formation of the endocardial cushions in conus cordis and truncus arteriosus. These cells, neural crest cells, are contributing in the formation of the endocardial cushions in conus cordis and truncus arteriosus. If something happens, some defect and abnormality is happening in the way of the migration of these kind of neural crest cells, there would be some problems or defects in the formation of this septum. Some heart defects may happen. So the neural crest cells also have some important role in the formation of the septum in the truncus arteriosus and conus cordis. And also this is one of the reasons for the transposition of the great vessels. They have role. Now that we can see the septum formation in the ventricles. Septum formation in the ventricles. As you remember, we had just one chamber with the name of left ventricle and the proximal part of the bulbus cordis, which was the right ventricle. Okay. One of the forms of septum formation was that in the one part or region or area of these chambers won't grow, but the other sides which located beside this region are growing so much. So this region will form a septum, a septum between these two chambers. The interventricular septum will form in this way. I mean, we have two chambers, left and right ventricle, in each side of the interventricular muscle. They are growing, and this part remains ungrown and forming the muscular part of interventricular septum. The ventricles are expanding, but the middle part won't expand. So it will form a septum between two ventricles, which is called the interventricular septum. The cells won't grow, and also the blood flow is entering to the ventricles, so they are expanding. They are expanding the ventricles, and the septum is forming. Sometimes, listen, the two valves of each ventricle won't fuse into each other, and there would be a cleft inside the muscular interventricular septum. There would be a cleft here. And some of the scientists call that the fifth place. So these two parts won't attach to each other. There would be a cleft between these two. Or maybe, for example, a cleft will remain here and the upper side of this septum. Okay, but the it doesn't have a name. It's just a small cleft between these two walls of each ventricle. Okay, so this is the muscular part of the interventricular septum. But the upper portion of the interventricular septum is the membranous portion. Longman. Longman is the reference for the embryology. The upper portion of interventricular septum is the membranous part. The membranous part of interventricular septum is formed by the endocardial cushions, which are placed in the atrioventricular canal, which are forming the atrioventricular valves, tricuspid and mitral. Okay? They are also growing downward and forming the membranous part of interventricular septum and fusing to the muscular part. And as I said, the neural crest cells are also contributes in the formation of the membranous part of the interventricular septum and the septum of truncus arteriosus and conus cordis. If the membranous part won't form, or for example a hole remain open between these two parts, we have VSD or ventricular septal defect. I will explain it. Any question about the septum formation in ventricles? Okay. Now about the semilunar valves formation. You know that semilunar valves are located at the beginning part of aorta and pulmonary trunk. At the beginning of aorta and the pulmonary trunk. When the partitioning or the separating of the truncus arteriosus is complete, listen, when its separation is complete, some semilunar valves become visible or appear in some trabeculate forms. They are become visible in some tubercles, some like tubercles in the truncus swellings. We have the truncus swellings and we can see these tubercles. Each part of the truncus arteriosus, I mean aortic part and pulmonary part, has their own tubercles. Each part has their own tubercles and they are growing. Okay, look at here. Each one has one tubercle, and here we have also some common tubercles for each one, for aortic and pulmonary ones. These tubercles are forming, I'm slide number 21. These tubercles are forming semilunar valves. Also, neural crest cells have some role in the formation of these valves. by the blood flow, these tubercles will form the semilunar valves. Look at here, the mesenchyma of semilunar valve. Here in the aorta and the pulmonary artery, the mesenchyme of these tubercles are proliferating and forming some valves, semilunar valves. Look at here. At first, these tubercles are in this form, then the mesenchyme will degenerate on the upper side of each tubercle. So, at last, these semilunar valves will form. We have the generation in the upper side of each valve. So we can see 3 semilunar valves at the beginning of the pulmonary artery and aorta. Any question about the valves? The function of the semilunar valve, we have talked about them. They prevent the blood flow into the right ventricle and left ventricle from the pulmonary artery and aorta. They have also a role in the formation of the semilunar valve. They help the endocardial tubercles to form these valves. Yes, it is important, clinical correlations. I will explain the clinical correlation of this part next time, I mean tomorrow, because our time is over. Do you have any question about the formation of the septums and the valves? They are so important. Okay?